sensory input or the activation. This is, a neuron has a unique characteristic that allows it to function. It shares that characteristic with a muscle cell. One word describes my youngest son when he wakes up in the morning. Someone this morning said conductive, and I was like, yeah, but irritable. Irritable means that it can respond to a stimulus, okay? Typically by opening or closing ion channels. So the dendrites and the cell body are irritable areas of the cell. Having the dendrites increases the amount of surface area that a neuron has. So this would be a dendrite. And then that surface area is expanded by more branches known as dendritic spines. And then the cell body would again have this very prominent nucleolus. So before we move on to the contents inside the cell body, I'm going to draw a larger view of one of these dendritic spines. And within the membrane of the dendritic spine, it has a receptor. And that receptor is an ion channel. All right? I'll draw another one over here. So notice that I gave this receptor a specific shape. That shape will recognize a chemical. That chemical might be acetylcholine, it might be dopamine, it might be serotonin, and those are different names of neurotransmitters. Right? And so if this neuron or another neuron releases a molecule that fits within that site, a neurotransmitter, then that channel will open and allow ions into or out of the cell. So this is identified as a chemically gated channel, or our fancy word is a ligand or ligand. Gated channel. And so that will control whether ions go into or out of the cell. If positive ions are flowing into the cell, we have a flow of charged particles. Guess what we just created? Electrical energy. Okay? So this is how the cell is stimulated. And we have these voltage-gated channels on the dendrites and on the cell body primarily. Did I say voltage? Ligand gated. So the cell body and the dendrites are the receptive area of the cell. Now, these neurotransmitters that are released are often proteins. So what are the cell organelles that are used to synthesize proteins? Are these proteins going to stay in the cell? Or are they going to be excreted from the cell? They're going to be excreted because they have to leave one nerve and go to another nerve to bind to that nerve's ligand-gated channels. So what kind of organelles do we use in the translation of messenger RNA for proteins? Ribosomes? And those ribosomes attach to what organelle that the new proteins are going to be excreted from the cell? Ribosomal or rough ER. All right, so a neuron, just constantly making neurotransmitters, is going to have a lot of ribosomal ER. In fact, this was one of the early sites where it was described by a scientist known as Dr. Nissl. And so these clumps of ribosomal ER are also known in a neuron as Nissl bodies. NISSL. And then, of course, we're going to have Golgi apparatus, because that has to process it. And leaving the Golgi apparatus, we have then secretory vesicles containing the neurotransmitters. Yes? Oh, I thought you had a question. So the neurotransmitters now are going to be transported down the long process, which is known as an axon. And the axon tapers from the axon from the cell body, and this tapering region is known as the axon hillock. The axon can be short or it can be very long. The nerves that innervate the muscles of your bottom of your feet leave the spinal cord at about the first or second level of your lumbar vertebra and travel all the way down your lower extremity to the bottom of your foot. So depending on how tall you are, that could be a foot to 5 feet. So many of the neurons that were studied for electrophysiology came from squid that had 20 to 30 foot tentacles. That's how long those neurons are. You have to go from the spinal, the squid central nervous system processor out to the squid, out to the tentacles. So this axon extends to another neuron or to a muscle, and it has multiple branches that contain these secretory vesicles. And these are known as axon terminals, and the swellings are synaptic bulbs or synaptic boutons. You're tired of drawing this. Stop. So, axon terminal. Synaptic bulb or bouton. So they're going to contain within them, and this is out of proportion size-wise, they're going to contain within them secretory vesicles, and the secretory vesicles contain the neurotransmitters. Now, how does that secretory vesicle get from here all the way down the axon? It's a more active form than that. So remember when we talked about cytoskeletal structures? We saw an image of a secretory vesicle with little walking proteins that use ATP. So what were these cytoskeletal structures that the walking proteins moved along? Yes. Tubules, but what? No? What were the hollow tubules that we find in cilia? The ones that are in motile, flagella, cilia, 24 nanometer in diameter, formed of tubulin proteins that form a hollow. But they form centrioles, microtubules. Microtubules. So the axon is going to be filled with microtubules. All right, transporting these vesicles along. And this requires energy, so what else are we going to have? Mitochondria. You're using up a lot of ATP. Okay. Teledendria and axon terminals are interchangeable. Okay, so you can use either term. So the appearance of a sensory neuron is different, but the same internal structures are going to be present because it's doing the same mechanisms of impulse transport. Yes?
On your picture, what is the neurotransmitter?
The red dots?
Those aren't the secretory vesicles?
The secretory vesicles are
The black circles
The black
around the red
circles.
dots.
all right, so now that we have a neuron up on the board, let's look at the fifth neuroglial cell, which is how this creates the myelin sheath of a peripheral neuron. All right, and this is known as a Schwann cell. Neurolimocyte is another name, but it's just too much trouble to say. So I leave it as a Schwann cell. Can I erase this drawing? Okay.
Once semester, we should really take a picture of all our images and like make a book or something. Oh, it'd be a lot. It'd be pretty cool. So,
The axon terminal are the branches off of the axon. The synaptic bulb is the swelling at the end.
so the telodendria.
Teledendria is the same as axon terminal.
Oh, okay, so it's okay. I said no.
Telo means end. So these are kind of the dendrites at the end of the axon is how what it's referring to. Yes.
So the neurotransmitters are the